Hello students, in today's lecture we are going to study about spermatogenesis. In previous lectures we have studied about oogenesis, and in this chapter we are going to study spermatogenesis. Now spermatogenesis is the production and maturation of male gametes called sperms. On daily basis around 200 to 300 million sperms are produced in male gonads called testes. Before we proceed further, let me recall you about the difference between oogenesis and spermatogenesis. Oogenesis was a process which was continued even before the puberty, while spermatogenesis is a process which only initiates at puberty. Now, what's the process at puberty is that FSH and LH hormone surge happens. This huge amount of FSH and LH hormone acts on the site of spermatogenesis called testis to initiate the process. Now this is the anatomy of testis. You can see that there is epididymis and there are these structures which are called seminiferous tubules. These structures are this structure is a tubular structure which contains of a loop of a lumen and an external wall. The walls are actually the site where the spermatogenesis is going to take place. Okay, so talking about the process, there are basically two phases of this process. The first phase, which is known as spermatocytogenesis. In this phase, primordial germ cells or the spermatogonia which are present in the seminiferous tubules, they are differentiated into spermatids. The spermatogonia are the diploid and the spermatids are haploid in number. Following are the steps of spermatocytogenesis. First of all, the primordial germ cells or the spermatogonia, which is diploid in number, is converted into primary spermatocyte type A by the process of mitosis. And then the primary spermatocyte type A undergoes meiosis 1 to produce two secondary spermatocytes. The two secondary spermatocyte then undergoes a second meiosis 2, to produce four haploid spermatids. These are the haploid spermatids, and then the second phase occurs. In the second phase, there is actually no division happening, only maturation of the previously produced spermatids. The spermatids are immature while it differentiates into mature sperms. Okay, following are the steps for the spermiogenesis. The first thing happens is the formation of acrosome. Acrosome is the structure which encircles, or we can say that it surrounds the central nucleus, and this is formed at first, and then the tail is produced. The tail is formed by centrioles. Centrioles elongates and forms the tail. And the third thing which happens is the shedding of residual body, shedding of cytoplasm indeed. Okay, what happens is that the excess cytoplasm is shedded, and all of the remaining structure which is remained in the end is now a mature sperm. This is the final look of the mature sperm. These mature sperms are ready, but they are immotile. They goes through another process which is known as spermiation. What happens is that they are produced in the wall of the seminiferous tubules, and what happens is that they migrate towards the lumen of the seminiferous tubules. They enter the lumen and spend some days in the epididymis of the testis, where they gain the motility to necessary for fertilization. They are ready to be then they are ready to fertilize the egg. Okay, so this was all of the process for spermatogenesis. It was consisting of two processes: spermatocytogenesis, this was all the process, and then there is another phase which is spermiogenesis, where no division is happening, and this was all of the process. Hope you understand today's lecture. If you understood, please make sure to subscribe our channel. Thank you very much.